So, as we're talking about protein folding and structure and how it's important for a protein's function, I'm hoping that somebody is asking, does it really matter if one amino acid in there somewhere is kind of out of place, or you swap out one amino acid for something else? Is it really going to change the shape and ultimately the function of that protein? Well, the answer to that is in some cases, yes, absolutely, it matters. If even one amino acid is not where it's supposed to be, it can create a whole world of problems for that protein, for that organism. And one example here shown on this slide is sickle cell disease. This is an inherited blood disorder that results from a single amino acid substitution in this hemoglobin protein that we've been talking about. So individuals that have sickle cell, they inherit a mutated version of the gene that has a single mutation that substitutes one amino acid for another. Just one amino acid in this massively sized protein. And in fact, it's only a mutation in one of the two versions of this, the subunits. So let's take a look. On the top here, hemoglobin, and the bottom here we have your sickle cell version. So when you look at the primary structure, the sequence of amino acids, and you look at the sixth position here in the normal hemoglobin polypeptide, you've got a glutamic acid in the sixth position. But those individuals with sickle cell actually have that glutamic acid replaced with a valine. And what this does is it causes the polypeptide to fold a little bit differently. So initially, it's not too much of a difference, still a globular protein. But when this beta subunit aggregates with the other subunits to form the full hemoglobin 4 subunited protein, it still forms properly. So the normal hemoglobin looks like this, sickle cell looks like that. You still have all 4 components put together. But because the beta subunits have that slightly different shape, they don't, in sickle cell, they don't kind of float around by themselves like they would under normal conditions. So what you see here is each individual hemoglobin protein is kind of doing its own thing. In sickle cell, because of that shape change, the protein starts to aggregate and it starts to create these really long fibers. And the really long fibers accumulate within the cell and they actually distort the shape of the cell. So instead of being a nice concave disc for the red blood cell, as you see under normal conditions, these fibers cause the red blood cells to take on a sickle-like shape, hence why we call this sickle cell disease. This shape prevents easy access or easy passage for these red blood cells through really small capillaries. They tend to get stuck. So this decreases, blood flow. Also, just intrinsically, the shape change for the protein decreases its ability to carry oxygen as well. And so this leads to a lot of issues for people that have sickle cell disease, especially when they are under stress or when they exercise. They may not be able to exercise because that requires a lot of oxygen to be pumped throughout their body, and their hemoglobin just doesn't have the oxygen carrying capacity. And so they suffer a lot of symptoms with this particular disease, especially under stress conditions. So yes, absolutely, a single amino acid difference can make a big change in the protein and its function and cause problems for an organism. Okay, so in addition to primary structure, right, we talked about how primary structure is going to determine how a protein folds, depending which amino acids you have where. Do you have a cluster of hydrophobic amino acids that are going to group together? Do you have a cluster of hydrophilic amino acids that are going to point out to the outside of the protein and contact the aqueous solution on the outside? In addition to the sequence of amino acids, this primary structure helping the proper folding of a protein, there are also external environmental chemical conditions that can affect the way that a protein folds. This can include alterations in pH, salt concentration, temperature, or other things. And what these other environmental factors do is they actually disrupt those bonds, the hydrogen bonds, the ionic bonds that hold the secondary, tertiary, and oftentimes quaternary structure in place. Once you start disrupting those bonds, the protein starts to unravel, and if it's starting to unravel, it's not going to be able to do its function anymore, and it becomes biologically inactive. And so the loss of a protein's native structure, this process is called denaturation. And theoretically, right, if you take a protein that's folded and you denature it, it should be able to refold. But many times within cells, this process is very difficult and maybe doesn't even happen at all. So in most cases, denaturation is kind of a permanent thing. You unfold the protein and it's permanently unfolded and permanently will no longer be able to do its job. Let me give you an example. A breakfast food that you may or may not like to eat that has a very high protein content is an egg. Right? So when you crack open an egg, it's got an egg yolk and it's got the egg white. The egg white has a lot of protein in it. Think about the color of the egg white and the consistency of the egg white when you crack it open, right? It's jelly-like and it's totally clear, right? You can see straight through it. Those proteins in that cracked egg are fully folded, right? They should be able to do their job. Now, what happens if you change the environment of that cracked egg? What if you crack it open over a hot frying pan? What happens to that egg white then? First of all, it changes color, right? It goes from being clear to being solid white. And also the consistency changes, right? It becomes a little bit more solid. What has happened is by adding temperature, heat, to those proteins, you have effectively denatured all of the proteins in that egg white and they've changed their shape. They can no longer function anymore. And this denaturation under normal conditions is permanent, right? You can't take that fried egg, put it back in the fridge where it gets cold, and it doesn't go back to being clear and jelly-like, right? You have tangled up those proteins so much that they can no longer refold and renature and go back to their original shape.